Welcome to CVL's virtual brown bag series. Let me take a moment to orient you to this virtual format. If you have questions, please type them into the Q&A throughout the presentation. We will make time to answer questions from the Q&A at the end, but we encourage you to ask your questions at any point during the presentation. Now I'm going to introduce our speakers for today, Meghan Swanson and Luke Moraglia. Meghan is an assistant professor here at UTD whose work focuses on language skill development in infants. She completed her bachelor's degree at the University of Wisconsin-Eau Claire before completing her graduate work at the City University of New York and her postdoctoral training at the University of North Carolina, Chapel Hill. She joined the faculty at UTD in 2019. Dr. Swanson will be joined by Luke Moraglia. Luke is currently a graduate student here working with Dr. Hervé Abdi on multivariate statistics, human memory, and decision-making. He received his bachelor's in neuroscience from Baylor University prior to joining us here at UTD. Thank you both for joining us today. The floor is yours.
Thanks to Dr. Seaman and Dr. Rugg for inviting us. Our talk today is a joint venture between myself and a talented doc student and Dr. Hervé Abdi's lab, Luke. This project is ongoing, and this is sort of the first time that we're presenting this work, so we're really eager to hear everybody's feedback. I'm going to start the talk today and lay our background and groundwork, and then I'm going to turn it over to Luke to talk about the details of the methods and results. Next slide. So as the title of our talk suggests, this is a talk about language skills during infancy. And the data that we are sharing are from the first 2 years of life. So our background will likewise focus on that time period. So infants are really on this path to developing language long before they say their first words and really at the earliest stages of life. Right around 3 months of age, infants begin babbling. They say their first word around their first birthday. And then sometime after that, around 18 months, we start to see what is called the language explosion. And this is a time period where infants are increasing the rate in which they're learning new words at a really remarkable rate. So by 2 years of age, many children say at least 50 words and use short 2 to 4 word sentences. But the question, of course, for a talk at somewhere like CVL is a neurobiological question, and that is, how does the brain support this developmental feat? Next slide. So infant brain development, especially in the first 2 years of life, is dynamic and rapid. During this time period, the infant brain, even just in sheer size, is remarkable in its development. So from birth to 1 year, the brain doubles in size. During this time period, neurogenesis and gliogenesis are starting to wind down, but synaptogenesis and myelination are really in full swing. Synaptic pruning is just starting kind of after the first year of life. So that is a process that's also involved, but not really a huge contributor, especially in the first year of life. Now we can measure brain development and brain morphometry by looking at the size of the entire brain, but we can also look at distinct aspects of the morphometry of the brain. And two of those are surface area and cortical thickness. So surface area is the area covered by the cerebral cortex. And if I can orient you to the figure on the right here, this is a coronal slice of the brain. And if you look at the red outline, that indicates the peel surface, and the yellow outline indicates the gray-white matter boundary. So in the studies that we're going to talk about today, the data that we've generated here, these are surface area measurements taken at this mid-cortical surface. So it's actually the midpoint in between the white and the peel matter. So if you took a dotted line kind of in between that that yellow and red line all the way around the brain, that's how we'd be measuring surface area. Cortical thickness is a quite different measure of brain morphometry, and that's the combined thickness of the neocortex, so that's 6 layers of the brain. And cortical thickness, if we go back to the figure for a second, if you look at the distance between the yellow line and the red line, that would be a measure of the cortical thickness. So we take these measurements at multiple vertices across the brain, and then from there you can generate measurements of surface area and cortical thickness based on the entire brain size or within specific regions of interest. So studies in adults have indicated that these two measures of cortical thickness and surface area are genetically independent. And the thought is that surface area is determined by the number of cortical mini-columns in the ventricular zone. This would be the radial unit hypotheses. So in this hypothesis, it's really the neurogenesis that's contributing to increased surface area. However, recent studies by infants by John Gilmore at UNC and in youth by Armin Rasnahan at NIH have found that there's more genetic overlap between surface area and cortical thickness than previously thought. So these are two distinct processes that are happening, but they're not quite as independent, especially earlier in life, than what was initially hypothesized. So studies in infants have found that surface area is actually highly heritable, while cortical thickness is markedly less so. So these research is based on twin studies. And those same studies found that neonatal surface area is influenced by sex and birth weight, whereas neonatal cortical thickness is impacted by environmental factors, including parental education level and maternal ethnicity. So the contributing factors related to these measurements of surface area and cortical thickness do tend to be distinct in some ways, but still similar in others. Next slide. So if we look at how surface area and cortical thickness grows and develops over the first 2 years of life, what we find is that these two things do have distinct and heterogeneous patterns of development. So the figure on the right is a slide from Lyle. This is work out of John Gilmore's group at UNC, and I apologize that it's a little blurry. I wasn't able to find a higher resolution figure for you all to look at. And what you can see here is if you look at the heat map for percent change, you can see that there's regional differences in cortical thickness and surface area growth. But what you can also see here is that the majority of cortical thickness growth is actually complete by 2 years of age. So by 2 years, cortical thickness is 97% of adult values, while surface area is 69% of adult values. So the thought is that cortical surface expansion accounts for most of the cortical growth after 1 year of age. I mentioned that there are some differences in kind of the regional patterns of surface area and cortical thickness. The kind of highest level synopsis is that primary and secondary sensory cortices tend to be slower growing, while regions involved in speech and language and some higher order associations, including the insulin cingulate cortex, were faster growing. Next slide. So I've just spent the last few minutes telling you the story about how both language development and brain development is undergoing substantial dynamic development, and the brain is highly plastic during this time period. And I think we actually have a pretty clear understanding of the development of the morphometry of the brain and the cortex during infancy, but we know far less about structure-function relationships during this time period. So there's some lesion studies that have been done in children, and they indicate that lesion site and size do not predict child's language performance in the same way as they do for adults with focal lesions. So children with perinatal focal brain injury often go on to have actually normal or near normal language skills. So this is really evidence of this early plasticity in the first early years of life. Now, this question of kind of creating a language neurobiology for infants is made more difficult by some limitations related to the technology. So we can't do task-based fMRI studies in infants because scans are always done during natural sleep, right? So you can do passive listening tasks, but you can't have infants in a scanner looking at any sort of stimuli or responding to any sort of stimuli in a way that is anything other than what they're doing while they're naturally sleeping. And folks really aren't using transcranial magnetic stimulation, or TMS, to do these reverse lesion studies in typically developing infants. I think there is some initial safety concerns with that, although I will say that there is some recent work that's been published looking at using TMS in perinatal brain injuries, so in infants. So we may find that as more safety information is available, we may learn some things about early language neurobiology using that technique as well. So the task of creating a neurobiology of language for infants is no easy feat. I put up this quote here because, for one, it's one of my favorite quotes. It's over 100 years old from the father of modern neuroscience, Ramon Cajal. And the quote is that the brain is a world consisting of a number of unexplored continents and great stretches of unknown territory. And I think at the time that quote held true for what we know about kind of the human brain writ large. But we've certainly learned a lot, especially in the last 20, 30 years with the advent of MRI imaging and other imaging modalities, about how the adult brain is set up and some of these areas of specialization. But I would actually say that that this quote still holds true for the infant brain. There's just a lot that we don't know about how the early brain develops. People have only been doing these studies, MRI studies on infant brains for the last 10, 15 years, so we don't have as much of a history, and then there's the limitations that I brought up previously. So while the goal is to develop mechanistic linking hypotheses that connect well-defined language skills to well-defined brain regions and neurobiological mechanisms, we're a long way from reaching that goal. And this study is really just one small step in that direction. But just because there's a lot that we could still learn about the infant brain doesn't mean that there are not theories that can help guide our efforts. Next slide. Thanks. So I'm going to share with you two kind of theories of brain development. The first one is what I'm calling here the maturational viewpoint. And this is the idea that the relationship between structure and function, so which parts of the brain perform particular tasks, is static, and that cognitive skills come online as the cortical circuitry intrinsic to that particular task matures. So under this framework, an area is said to be mature when it achieves an adult state like functioning, and any other sort of state of functioning is then deemed to be immature. So there's some initial appeal to this approach. It would be great if we could just downward extend models developed into adults into the infant brain. But this model doesn't fully explain some initially surprising observations on especially functional brain development. So for example, recent evidence suggests that some of the regions that are the slowest to develop by neuroanatomical criteria, so regions like the prefrontal cortex, can be activated from shortly after birth and appear to mediate cognitive function. We know this also from some of the landmark studies that were done in neonates looking at functional, so this is passive listening task, and what they found was there was activation in the paracipient network, these kind of putative language regions, long before infants were talking themselves. So it's clearly not the case that a brain region needs to be fully mature before those regions can be activated to help support language. So I think that this maturational viewpoint is not a great model for which to explore infant language neurobiology, but there are models of adult language neurobiology that are potentially also helpful. Next slide. And like I said, if we were following through with this maturational viewpoint, we would take these adult models and just downward extend them into the infant brain. So I don't need to explain this classic model of language neurobiology to this group. It's been around for 150 years, and it's in all the textbooks that we read and learn from and that we teach to our students. So in this framework, Broca's area is related to speech production at its kind of most basic level, and Wernicke's area is related to speech understanding. Those two regions are connected by a white matter fiber tract called the arcuate fasciculus. But this model is problematic. It is anatomically underspecified, and it underestimates the number of brain regions involved with language. So two researchers, Tremblay and Dick, asked a group of scientists, and I just think this study is kind of very clever. They asked a group of scientists, the majority of which held PhD, and reported that they had expertise in neuroanatomy to indicate the regions that were part of Wernicke's area. And what the figure on the right is showing you is the different potential regions that were selected by the researchers. And you can tell that even amongst these group of people that mostly held PhDs and had knowledge about neuroanatomy, there was no conclusive conclusive finding on what is the neurobiological boundary or the anatomical boundary of Wernicke's area. So this model lacks anatomical specificity. And the other issue I brought up was that it underestimates the number of brain regions involved with language. And more modern models of language neurobiologies have begun to address that specific criticism. Next slide. So the figure on the right here, and it's probably a little small for you all. It is a complex model. This is the dual stream model of language neurobiology brought forth by Greg Hickok and David Poppel. And the model is certainly more complex than the classic model, and you'll notice that it also includes the left and the right hemisphere. But this model, again, is based on the adult brain. So the question remains, are adult models appropriate for the infant brain? And this model includes a ventral stream and a dorsal stream. And I'm not going to get into the details here, other than to say that this is a model that certainly exists, includes more anatomical regions, but once again is based on adults. Next slide. So an alternative approach to thinking about language neurobiology and how you extend this down into the infant brain is to think about the theory of interactive specialization. And this was put forward by Mark Johnson. And under his framework, the brain is not built via pre-specified genetic blueprint, right? So rather, brain development is complex and dynamic and back-propagated interactions between genetics, brain, body, and the environment. Other key features of this model include the idea that developmental change in cognitive skills or behavior will be accompanied by widespread changes across multiple brain regions, and that the activation of networks of regions will, as a whole, become more selective to given stimulus or task context during development. So in this theory of interactive specialization, you don't start off with a specialized brain, but rather you get to a specialized brain through activity-dependent neuronal processes. Next slide. So I've spent, this time so far talking about early language and neurodevelopment, and have talked about this kind of in general terms, in terms of typical development or normative development, but this is also a talk about autism. So before we kind of get back to the data and back to these models, I want to spend a few minutes just making sure that we're all on the same page about some of the basics of autism spectrum disorder. So autism is a lifelong neurodevelopmental condition affecting currently the rates is 1 in 54 children. We know that autism is highly heritable, but there are many genetic causes. It's not a condition like Down syndrome, where there's a clear single genetic cause that you can measure in a genetic test. There are many different kind of genetic pathways to autism, and no single pathway accounts for a significantly large portion of the pie. Core features of autism include deficits in social communication and interactions, and the presence of repetitive and stereotype behaviors. So you'll notice here that there's sort of two aspects here. So it's deficits in social communications and interactions. This could be how an individual interacts with other people, could be their use of things like gestures and joint attention. And then the interactions piece is meant to get at sort of that dyadic piece of how these children socially interact with their peers and their parents and everybody else that they encounter. The other side is actually not a deficit, but rather a presence of something. So this is the presence of repetitive and stereotype behaviors. These could be stereotype movements, the classic ones being hand flapping. It could also be restricted interest, where a child has a favorite topic that they like to discuss or one game that they like to play. But it could also manifest in children, having difficulty veering from schedules and routines. So this presence of repetitive and stereotype behaviors is actually quite complex, and it's not a monolith in any sense of the imagination. So the current average age of diagnosis in America is currently just over 4 years of age. There are some differences depending on the severity of the child and the age that they're picked up. So but you can actually have a clinically reliable diagnosis at 2 years of age. Those clinically reliable diagnoses are done by people that have expertise in autism using gold standard criteria, so not necessarily the way that most children in the community are assessed. We do know for autism that getting into early intervention, which is treatment from birth to 3, leads to improved outcomes for these children. But if you're thinking about this timeline in your head, you may be wondering and realizing that there are a lot of children with autism who completely miss out on early intervention because they're not diagnosed until that time period is actually ended, right? So there's been a lot of emphasis in the field and attention in the field about how we lower the age of identification and entry into treatment. And to do that, you need to be able to study autism during infancy. And the way that you do that and solve that conundrum is actually a clever one, and it's utilizing a family design. Next slide. So this family design that I mentioned is the baby sibling design. Some of you who have heard my talks before are probably familiar with this design. So we know that 20% of infants who have an older sibling with autism will go on to have autism themselves. So there is a genetic liability for autism within these families. So the way that you study how autism develops and unfolds is actually by studying families. So you have a family that has a child with autism, and then they're having a subsequent child. We recruit that baby into the study when they're 3 months of age or 6 months of age. And then we prospectively follow them throughout their first 2 years of life. And we do this by creating groups of children. So these children that have an older sibling with autism are what we call high familial risk infants. So they have an older sibling with autism. And as I said, about 20% of them go on to have autism. 80% of them will not meet diagnostic criteria for autism. We also have a control set of infants in the studies that we do, and this is all work through the Infant Brain Imaging Study, or the IBIS network. And the control group are what we call low familial risk families. So these are babies that have an older sibling, but that older sibling does not have autism. And we see what we would expect from the population in these kids. So about under 2% of the low familial risk infants will go on to have autism themselves. We actually exclude those kids from all analyses because the sample size is too small. But then the overwhelming majority of those kids, 98% or over 98%, are actually negative for autism. So they really serve as a community control group. Next slide. So I'm not going to spend a long time talking about language and autism, but just to mention that while language is not a core feature of autism any longer, it is something that is a challenge for a lot of children. So at a group level, kids with autism are late to meet early milestones. At a group level, they also score lower on standardized language assessments at 12 months, and that group difference is more pronounced at 24 months. There's a substantial proportion of these kids that remain minimally verbal, and the kids that do have language skills really have an array of difficulties in speech production, comprehension, or pragmatic language. So language is not a core feature of autism, but it is something that impacts many of these children. Next slide. So the aims of our study are really threefold, and the first one is to see if surface area and cortical thickness measurements are related to later language skills. We then asked a developmental question of, do these patterns of association change between 6 and 12 months of age? And then the last question is sort of a descriptive question at this point in time, asking the question that, do these associations actually differ based on the infant's neurodevelopmental status? Next slide. So we get these scans, as I mentioned, during natural sleep. The scans were collected at 4 different sites using identical scanners. We collected the data at 6 and 12 months, and we used the T1 and T2 scans to generate global brain volume and cortical measurements for surface area and cortical thickness. And at this point in time, I'm going to pass it off to Luke to get into more of the details of the methodology and the results.
All right, thank you. Trying to do the PowerPoint and the live stream at the same time. So just want to make sure it's all going good, but it looks good. So thank you, Megan, for that, and Kendra earlier for your introduction, and everyone here at CVL for having us. We're very excited to be here. So I'm going to talk just a little bit about the current data analysis that we did. And so just to start off, giving you guys a description of our data. So like Megan mentioned, we have 3 groups that we analyzed: the low familial risk, negative for autism, and then also the 2 high-risk groups, negative for autism, and then those that do eventually go on to develop autism. And I'll talk more about the analysis method in a second, but we did 3 separate analyses and did them just by group because we really wanted to look at what are the trends within that group as they relate to these language measurements and these brain measurements. So looking at our variables of interest here, we had 78 brain regions that are defined by the AAL Atlas. And for each of these brain regions, we had the 4 measurements, so surface area and cortical thickness at both 6 and 12 months. For our language measures, we used 2 subscales from the Mullen scales of early learning, the receptive and expressive language subscales. And these were measured at 24 months of age, so getting close to where you're having more language development in kids, and then also closer to that reliable diagnosis for these kids. And throughout these analyses, we controlled for gender, mother's education level, the data collection site, and then also the age at the brain scans and Mullen administration. And just, you know, we're saying that these were at 6 and 12 months time points, but there is some variability in that. You know, these babies are not scanned exactly on their 1-year birthday, and just because there is such rapid change at these ages, we do want to make sure that we control for even small variations in the age of the participants. So how did we analyze this data? We used a multivariate method called partial least squares correlation, or PLSC. This is a method that aims to analyze the relationship between 2 separate quantitative data tables. So in our case, we have one data table that contains the surface area and cortical thickness measurements, the brain score table kind of. And then we also have another that contains those 2 Mullen language subscales. And what this analysis is trying to do is say, okay, well, if we look at the correlation between these 2 tables, like you see in the correlation matrix at the bottom of the screen here, you know, we have this pattern of correlations. What are the most salient, important patterns in that very complex correlation matrix? So the goal is to identify the variables that are most contributing to this overall pattern of correlation that we see. And if you're familiar with principal component analysis, this is akin to performing a PCA on this correlation matrix. So just trying to extract those trends that best explain the matrix here. We can see which variables contribute most to these patterns by looking at their loadings in the results. And we assess which loadings are reliable from a statistical inference point of view using a cross-validation measure that's based on bootstrap resampling. Just to summarize all of this here today, the regions that are related to language do differ between the groups. We saw a wide variety of regions that contributed to one and not the other. And then also the surface area and cortical thickness just don't measure the same thing. They're not just a measure of how big the brain is. They're actually really very different things. And then within each group, you can see the different trends there that we saw. Also, the stability of these areas really does vary from 6 to 12 months. Some groups of regions are really stable from 6 to 12 months, and that actually gives them maybe a little bit more credence as being important and kind of catches your eye a little more. But it is important to kind of look at all of these. So I think at this point, I'm going to just briefly turn it back to Megan to kind of wrap us up on this talk. So Megan.
Thanks, Luke. I think I remembered to unmute myself this time.
I hear you.
So as Luke just showed you this data from this kind of initial pass of results, what we're finding is that we don't really support this maturational viewpoint of language neurobiology. And this data has much more support for something like the theory of interactive specialization, and that the brain is just not fully specialized during infancy, and it takes a while to kind of get there. We do have kind of future directions that we're thinking about. Even within this set of data, there's additional analyses that we're moving towards. We showed you data from 6 and 12 months separately. We do have the desire to actually look at longitudinal differences, and we also have brain scans from these kids at 24 months of age, so we can actually create longitudinal trajectories of surface area and cortical thickness across that time period. And then the other thing that we want to do is actually measure these group differences. So we talked about them descriptively in our talk today, but we can do additional tests where we actually say, are these patterns of association different amongst the different groups in our study? So we have much more work to do on this data set. That's certainly the case. But we also kind of have like higher level goals that we're working towards, right? So how do we move towards these linking hypotheses? One thought that, we always like to talk about is replication. We need to replicate these results. In some ways, this is a little bit unsatisfying because I don't know how much it really moves us forward closer to linking hypotheses. The other thing to keep in mind is that there's a preprint that's out there by a bunch of eminent scientists saying that these sorts of whole brain behavior association studies, you really need sample sizes north of 2,000 in order to have stable brain behavior analyses. So it may be a while before we can truly replicate and have stable results for these sorts of brain behavior association studies. It may be the case that we need to move to other methodologies in order to make steps towards these linking hypotheses. So I think MEG is a great example of a way that we could overcome some of the limitations of MRI imaging during the infant brain. So MEG has good temporal and spatial information, and children can move their head and be awake during the image acquisition. So our options for how we could test early language acquisition and language understanding really are opened up a lot using this technology. And then last, I put on here this idea of a natural intervention study. So a lot of my work looks at how the language environment or caregiver speech supports early language skills, but we may actually learn something about language neurobiology if we look at differences, individual differences in caregiver speech and see how that is impacting brain development in the infants. And of course, just to keep our long-term goal in mind for us and to let you know where all this work is kind of headed, we really want to, inform personalized medicine approaches to early intervention for these infants. So in the perfect world, we can use the brain or proxy measures for the brain to figure out which kids need language intervention, which kids need some sort of sensory intervention, or which kids need an intervention related to social cognition and joint attention, for example. So that's, a long-term goal and something that we always keep in mind, but it really is the reason why we aim to do all of this work. Next slide. So I think with that, I'll wrap up, and we have plenty of time for questions. I do just want to take a second to acknowledge the families that participated in this study. I didn't go into kind of the nitty-gritty logistics of the IBIS network, but these families are from across continental United States, and they get on an airplane with oftentimes their child with autism, their brand new baby, and they fly to one of these clinical data collection sites, and they trust us to put their babies in the scanners. And then they come back and they do that 3 or 4 times before these children are 2 years old. So it is a remarkable commitment that these families have to the studies, and I think that needs to be recognized here. And then this work, as I mentioned, is part of the infant brain imaging study, or the IBIS network. My lab has also contributed to this work, as have other groups in the ABD lab. So our funding is on here and our contact information is. And I think with that, we'll take questions if there are any.
Great. Thank you guys again for coming and giving this presentation today. We have a question from Mick to kick us off. Are the brains analyzed in native space? And if so, did you control for individual differences in overall brain size? And do reliable templates exist that allow infant brains to be normalized to a standard space?
A lot of fantastic questions, Mick. Luke, can you cue to my image processing detail slide? Thanks. So full disclosure, I process the diffusion data for this study, and I'm kind of an educated end user of the structural data. So this image processing pipeline used Civet. Our image processing cores for our structural data are led by Alan Evans at MNI and Martin Steiner at UNC. So my understanding of the Civet pipeline is that it starts with a linear or affine registration of the native images to stereotactic space using a model. And the models that we use in our studies are all study-specific models. So that first step is that the images are registered to our study-specific atlas or model. So that probably gives you a hint at one of your other questions, that do reliable templates exist that allow the infant brains to be normalized to a standard space. So there's not standard templates that are out there that are publicly available like there are for the adult brain, and you cannot use the adult brain atlases in the infant brain. It doesn't work. The infant brain is just fundamentally different. So, if you see that happening, it's not a good thing. We have in the IDIS network, we almost always create study-specific atlases for if we're looking at, subcortical volumes or our DTI data or our structural data or even our functional connectivity data. We are working to make some of those atlases publicly available so other groups can access them. But no, it's not the case that there are reliable templates that are kind of publicly available. At this point in time, our data set is so big that we kind of have our standard templates that are based on our previous data, and we just fold in new data and register it to that atlas. And then there was a question about individual differences in overall brain size. And I'm hoping Luke can remind me, because we had a lot of conversations about this and the different iterations of this project and what sort of brain size we should be controlling. Should we be controlling for total surface area or intracranial volume or total, total tissue, or what we should do? And I honestly can't recall what we ended up doing. So I'm gonna punt this one to Luke.
No
to
problem.
mention.
Yeah. So in this particular iteration that you're seeing here, we actually didn't control for any sort of the total brain volume or intracranial volume or anything like that. That is something that we're still working on and considering, so good question to ask. And part of it for us was that we wanted to kind of say, well, we're going to take this exploratory approach anyway. Maybe there is some aspect of the brain, the actual surface area sizes that within a group are interesting to look at. So
I think the thing that we struggled with, Mick, is also just thinking about what should the research question be. So should the research question be about sort of relative growth in one of these surface areas regions, which then you would want to be controlling, or if we were interested in absolute growth. And so we took this first approach, and I know we have kind of done analyses controlling for different things at different times, but this initial approach does not control for total brain size. It's just about the absolute growth in these regions or the absolute size of these regions. I think that will be much more important when we start to actually compare the different groups, because we know that there's differences in brain sizes in our autism sample.
I just read through Gagan's question here, asking if we could talk about the morphometric differences between the groups, those that express ASD versus those that don't. And this is something that we started to touch on a little bit. And here's a really good figure from the Hazlitt 2017 paper in Nature. So there are some differences between these groups in overall surface area and in regional surface area as well. So these are some groups that, these are some regions that actually show significant differences between the low risk, typically developing groups and that group that goes on to develop autism in the hyper expansion of surface area. So actually the change in surface area from 6 to 12 months. Overall, the main effect being that these kids do have larger surface areas overall throughout the brain and in total surface area and total brain volume and things of that nature. Also looking at main effects of the language production and reception in the data set, these kids do have lower scores overall. And I'm not speaking any sort of statistical test way, so don't ask for a p-value, but just overall the trend is that they have lower scores on this Mullen assessment as early as 2 months. And actually, that if you wanted to try to predict group membership with some of these more machine learning type algorithms that try to classify your groups, the best data that classifies them in this sample are actually the Mullen early language scores and not their brain surface area or cortical thickness. So the part of the motivation to really look at the surface area and cortical thickness is because you can get those data at 6 months or 12 months, so earlier on, versus by 24 months, if you're testing them on this language measurement, then at that point you might already be able to make a diagnosis anyway. So with the kind of overall big picture goal being earlier diagnosis, that was part of the motivation to look at these earlier time points.
Yeah,
so
thanks, Luke.
And answered all that question. Mm-hmm.
And I'll just add to that, Gagan, that we've actually published on the language, the longitudinal language data for this sample. And as Luke said, they score lower. So the groups don't differ at 6 months, they score lower at 12 months, and then the group differences are enlarged by 24 months of age. There's some measurement issues involved in measuring language production in 6 months and 12 months old, but it is the case that already at that 12-month time point that they're scoring lower than their peers.
All right, I think we have one more question from Shruti, and it's about the high-risk negative group, wanting to know if what your future goals are for this group. Are you going to look at brain differences in relation to outcomes for that group?
Yeah, that's a great question, Shruti. So, so in the paper that I published looking at language outcomes, we also looked at if there was a group of infants that had early signs of language delay. And what we do see, as Luke mentioned during his part, that these high-risk negative infants do have higher rates of language delay than you would expect in the population. So we've talked about creating like a language delay or risk of language delay group and using that as a way to subset the high-risk negative infants. I think we haven't quite decided on what the best approach is. The ends get a little challenging there because the sample size just gets small, because it's really only, I'm texting my memory a little bit, it's probably somewhere between 10 and 15% of the high-risk negative infants that actually meet that criteria for language delay. And then I think we're out of time.
Yeah, I was going to actually say thank you so much for coming. I'm giving you a round of applause over here on my end. I'm sure people are doing that at home as well. Thank you, and we'll be back in two weeks with our next virtual brown bag.